Alright, guys and gals, welcome back. Kember here. Using a makeshift microphone, should have one coming in the mail, so I hope the audio quality will be maybe better today. You never really know. But welcome back to Kember. Today I'm going to be talking to you guys about molecular rulers. And by that, what I truly mean is, what is ways in which we can measure stuff that is so unimaginably small that we can measure distances at the molecular level? And we can do that using a technique called FRET. But before we really go to how this technique works, we still need to have an overview, just generally, of spectra and just generally looking at a Jablonski diagram, which I'm going to present now. So what we're looking at here, the only arrows you really need to worry about are the green one and the purple one. So what first happens, this diagram is just showing the energy levels of some atom. So when we're starting at the very bottom with the green arrow, that is signifying an electron. And what happens with electrons is they can either sit at some ground energy state, or if they get enough energy, they can get promoted to a higher energy excited state. So a passing photon will have some given amount of energy based on the wavelength of that photon. If that photon or light that hits this electron matches the exact energy needed for that electron to go to a higher energy level, that's what it's going to do, and that's what's being shown in this purple arrow here. Once that electron is in this excited state, it's going to go through vibrational relaxation, where different vibrations and stretches of bonds or solvent quenching, other such stuff that happens in the molecule, there's going to be a loss of energy due to latent heat from that electron. And eventually, once you go through this vibrational relaxation, you end up at the bottom level, this excited energy state, and this electron is ready to drop back down to the ground state. And so when that happens, when you go back down to the ground state, you're going to re-emit another photon, but this photon will necessarily be redshifted because it had to lose some energy when it underwent vibrational relaxation. So that means the photon released by this electron dropping down back to the ground state will have less energy than the original photon, which excited the electron to get up into that excited energy state in the first place. So now what happens when you have two fluorescent molecules together, though? And that is really when the wacky stuff starts happening and when we can start talking about what FRET actually is. So let's say we have this first molecule, this donor molecule. As you can see from this graph, the blue dotted line is showing that this donor molecule absorbs light, a very specific part of the spectrum. It's going to absorb blue light and it's going to re-emit green light. But what happens if the emission from this donor matches the absorption of some other molecule that's nearby it? In that case, what you can have happening, as we can see with this yellow acceptor molecule, is used to absorbing yellow light and it emits red light. And so what happens in a FRET, which that stands for Forrester Radius Energy Transfer, as the name suggests, you have an energy transfer from this donor molecule to the acceptor. And this happens in a non-radiative way, meaning that no photons are exchanged between the donor and the acceptor. And this technically happens through dipole-dipole coupling, which is honestly a little bit in the weeds even for me, so I won't really mention what that is, other than when you have FRET happening, you're going to have this energy directly transferred to an acceptor molecule if you're close enough. And what this will seem like is FRET works on a distance of roughly 1 to 20 angstroms, kind of loosely 1 to 10. In this distance, if this donor and acceptor molecule are within 1 to 10 angstroms of one another, what'll happen is, let's say you shine this blue light on the donor, and these donors, the donor and acceptor molecule, are close enough to the point where they can have FRET happen. You won't see any of the green light emitted from the donor, and you'll only see the red light emitted from the acceptor. And so if you reverse the scenario where the donor and acceptor are now farther than 10 or 20 angstroms apart, what you're going to have happen is you're going to have the absorption of the blue light from the donor. It's going to re-emit green light, and then only some of that, you won't even have FRET occurring at that point, because then you're going to have a photon interact with the acceptor somehow, and you won't have that energy transfer happening. And this is basically the way that we can understand as to how far these two molecules are from each other, is using their Forster radius. So this Forster radius is specific for a donor-acceptor pair. So if you're using two specific fluorescent molecules, they're going to have their own set Forster radius. The Forster radius being at what distance from each other do these molecules transfer 50% energy successfully. Meaning, for example, if you have a donor and acceptor, we're at 15 angstroms, this donor will transfer its energy 50% of the time to the acceptor, then the Forster radius will be 15 angstroms. So then what you can use with this information is looking at the efficiency of this transfer. So as I was mentioning earlier, if you have a large distance between your two fluorescent molecules, you won't be able to have FRET. But when they're getting closer together, depending on the light that your detector is receiving, you will know whether you have energy transfer or not. And based on this equation I'm showing on the screen here, it's showing that the efficiency of FRET is simply equal to the Forster radius over the Forster radius plus the distance between that FRET pair that you're using. So as I mentioned, there are some limitations to this, namely the distance constraint, which is you need to be anywhere from 1 to roughly 20 angstroms distance, the donor to the acceptor, and that's in angstroms. So in nanometers, that's 0.1 to 2 nanometers. That is the effective range of small molecule FRET. And this also depends on the orientation of this donor molecule relative to the acceptor molecule. Also depends on the refractive index of the medium, the quantum yield of the donor, as well as the extinction coefficient of that acceptor molecule. And as I mentioned, the overlap of the spectrum. You want to have an overlap between the emission of the donor and the absorption of the acceptor. So I'm going to walk you guys through just a specific paper here by Sharona Gordon, Dr. Sharona Gordon at the University of Washington, and others. And their paper that they wrote is visualizing the conformational dynamics of proteins in solution at the cell membrane. And so specifically they're looking at the maltose binding protein, which let me walk you through what it does. Protein in the name, that means that this thing must be made up of the 20 canonical amino acids, right? Maltose, that's a sugar. So why would we be studying the movement of the maltose binding protein? Well, the main reason for this is obviously for human health. There's pathogenic bacteria, and this bacteria needs to get energy from somewhere, and it can get it from the maltose binding protein, which takes maltose from the outside of the cell and brings it inside. So it could potentially make a great target for antibiotics, but it's also important to study because I'm not sure if people are aware of this, but your body is filled with hundreds, if not thousands, of bacterial colonies which do molecular work for you and digest your food for you. So by knowing how this binding protein folds and interacts with maltose, we could get a better idea as to how we can use it to leverage the maltose binding protein as a antibacterial. We can use it to create new antibacterial drugs, or potentially give us a better insight as to how our own digestive properties works with our microbiome, I guess. But looking at the maltose binding protein itself, here we see this like cyan structure. That's the maltose binding protein itself. And you can see the maltose, that sugar there is shown in purple. We have these 4 sites where they're going to add fluorescent molecules so that they can use Fret to calculate the distance between these Fret pairs. So you have a Fret pair on top and on bottom. And they're also labeling the specific number or the position of where this Fret pair is going to be within the structure of the protein. So just to remind you guys, proteins are made up of the 20 canonical amino acids. So as I'm showing you here, there's these 20 where this maltose binding protein is roughly, I think the last slide has said that there's roughly like 350 amino acids connected together. So of these 20, you're going to use multiple of these 20 to make a 350 letter string of these amino acids, and it's going to fold into this maltose binding protein. And it's only showing the backbone structure. So if you're looking at this diagram again, you're only seeing the OH, the O, and the NH2. You're just seeing that, and all the functional groups are chopped off just for a little more cleanness to it. And so the way that this maltose binding protein is synthesized is that these amino acids, these hundreds of amino acids, are encoded in an mRNA sequence. So if you have roughly 300 amino acids in a protein, that means that you must have a roughly 900 base mRNA that it was synthesized from, because you need 3 bases to make 1 codon, which translates to 1 amino acid made in the ribosome. So the problem that these researchers had is that they wanted to use an unnatural amino acid as one of the fluorescent molecules. However, the body doesn't make this amino acid normally, so they have a really cool trick as to how they got around this actually, which I'm gonna show here. Was being shown is they wanted to use L-ANAP, which this is a fluorescent molecule which gets excited around 360 nanometers, I believe, and it emits light at roughly 490 nanometers. Okay, so basically the way that they repurpose the cell machinery to get L-ANAP expressed is that they're using 2 different plasmids, which are basically just ways to deliver a very specific sequence into the cell so they can get synthesized. In this case, one of our plasmids is called MBP tag, which that stands for maltose binding protein, and the tag signifies there's 2 different tag sites in the maltose binding protein. Those tag sites, the tag site refers to one of the stop codons, which if we remember a codon chart, there's 3 different stop codons. So when a ribosome comes up on a stop codon, it'll usually just stop ending. It'll just end that protein synthesis. Whatever has been synthesized so far, that's it. So basically what they're doing is they put a stop codon at the place where they want to incorporate L-ANAP. And then what they're doing is they also included this amino acyl tRNA synthetase and the tRNA itself, which this amino acyl synthetase, that's basically the enzyme that you use to incorporate the non-canonical amino acid. In this case, they wanted L-ANAP. So it incorporates that onto the tRNA, and this tRNA binds to the tag codon. So in this case, they're assuming that a different stop codon is being used to end the protein, and they're using one of the stop codons instead of for its normal purpose. They're repurposing it to express L-ANAP. And then they also had other changes in the maltose binding protein. They also added some histidines and cysteines, which allow for the binding of the Fret acceptor molecule. For the second molecule that they're using in this Fred pair, it actually isn't fluorescent at all. In this case, it's actually copper, and it acts as a quencher. So what you can see is that this copper is being held in between, I think those are histidines, I'm actually not super sure, or this other T-tac structure, which is just holding in this copper ion. And as I said, this copper ion is acting as a quencher. So what that means is if this LANAP is within 10 angstroms of the copper ion, you will have Fred happen. So if you excite LANAP with 360 nanometer light, you're going to have then an energy transfer from that amino acid to the copper, but then the copper won't re-emit it. It's just going to take it and not re-emit. So you're going to have darkness. So if you see this amino acid right nearby the copper, then you will have no light at all that is going to come out from this. However, if this amino acid is farther away from the copper, for example, let's say 15 or 20 angstroms away, then not all of this energy is going to be transferred, and some of it is going to be allowed to radiatively come out of this amino acid in the form of 490 nanometer light. And so this is the method in which they're measuring the distance between two various points on this maltose binding protein. And so just looking at the actual video itself, this is the final data they got, or little animation, where you can see the magenta in there, that's the maltose in the middle, and then they're measuring in angstroms the distance from those 2 points. And what you can see is they notice that this maltose binding protein acts kind of like a clamshell, where it closes onto the maltose binding protein before it gets sucked into the cell. As I mentioned, this has implications in antibacterial drugs possibly, as well as giving us some more insight into our own microbiome and how these bacteria that feed us actually work. There are other methods to look at these molecular type motions, for example, using NMR and other such techniques, but that is way above my pay grade, so I hope you enjoyed just a short video talking about just a cool paper, I guess. Yeah. And as always, I hope you've enjoyed. Have a good one.